In today's lesson, we're going to talk about what's actually part of the very end of our course, Unit 4, Area Study 2. But we're going to talk about it because it fits so well with the immunity topic that we just did. So it just really makes sense to talk about it now. And the topic we're going to talk about is the emergence of new disease. Okay, how new diseases actually come to be. Okay, so yeah, in this little comic it says, I know it's our job to reassure the public. Is anyone else going to acknowledge that this looks like the beginning of every zombie movie ever? And here we have it's the Ebola quarantine zone. All right, so like I said, very end of the book, so of the topic of the course, so it's the very end of the book. All right, 3 good questions here. Have a go at these, and we'll have a look at them inside of class. So in the study design, we're looking at this biological knowledge in society. This area of the study design is all about taking different concepts that you learn throughout Year 12 biology and actually applying it in terms of actual new human society, right? So we're going to talk about different strategies that deal with the emergence of new disease in a globally connected world, including the distinction between what we call epidemics and pandemics, and the use of scientific knowledge to identify the pathogen and different types of treatments for that pathogen. All right, so let's talk about the WHO and when they talk about the uncontrolled spread of disease. So when a certain infection disease initially appears in a community, we call this an outbreak. All right, let's say that here we have good old Cassidon. I represent Cassidon with a little red dot. Go away. And in Cassidon, there might be an outbreak of influenza, the flu. Okay, and so let's say a few hundred, maybe 500 people in Cassidon get the flu, which is devastating for a little town like Cassidon, right? And maybe even that flu is going to pass around to Hamilton and Mount Gambier and Edenhope. But we're just in our relatively small area. That means that we have an outbreak of the flu. But let's say that if that flu starts to actually spread around really uncontrollably within that community, nearby communities, but all inside of Australia. So let's say that the flu passes on to Melbourne and Adelaide and even maybe to Brisbane and, oh gosh, Perth. And in all over Australia now, there's a big flu epidemic, okay? Let's say a few of these people that have the flu get on planes and they start flying. They fly to New Zealand, maybe they fly to China, they fly all the way to the UK, and some people might even fly to United States. And what happens then? Now we have what we call a pandemic, which is when a disease now starts to spread like mad all around the world and might be the end of humanity. No, hopefully not. Let's look at some examples of pandemics and epidemics. A very classic epidemic that recently was in the news, and actually very more recently actually started to resurface a little bit, is the Ebola virus epidemic, where we had the spread in West Africa, across a few countries in West Africa, and with some very small isolated cases in the US, but it wasn't spreading inside of the USA. So this is an epidemic because we were actually really, the uncontrollable spread of Ebola was in West Africa, okay? And then we can have pandemics such as the influenza pandemics in 1918, the Spanish flu, which started in Spain but spread around all of Europe and much other countries around the world. In 1951, we had the Asian flu, same story. The swine flu, also in Asia in 2009. Okay, and flu is all an infectious disease caused by the influenza virus, as we know. And then SARS in 2003 is another interesting epidemic which took the lives of around 800 people in southern China, all right? The identification of the pathogen, the SARS coronavirus, and the sequencing of its genome all happened within the space of about 3 weeks, which is pretty amazing if you ask me. All right. We had very, very strict measures that were involved, such as quarantining the population, screening visitors, slow the spread of this new disease. And since 2004, we are happy to say that there have been not one new case of SARS, and that's all due to the amazing work done by these health professionals and biologists and immunologists especially. So how did we identify a pathogen like SARS? Is through very careful steps of different processes, okay? First of all, it's really important that we can identify a pathogen, because by identifying a specific pathogen, we know more about that pathogen, and so we can, one, stop it spreading, and two, start treating it. Okay, so there's different imaging techniques, all right, such as here we've got some electronic microscopy of specific rod-shaped bacteria. We'd be able to start then treating for rod-shaped bacteria. We can identify the antibodies that our bodies are producing. If we know the antibodies our bodies are producing, then hey, we know how to fight that disease, because we, one, know specifically what the antigens of that disease are, and two, our bodies are giving us a pretty good clue about how to fight it. We can directly analyze the DNA, which is something we're going to look at later on in the year. And then we can also do some more old school phenotypic techniques, such as the classic one is called gram staining. And that's done with different bacteria.